Okay, good afternoon. Thanks, Brian, for a nice introduction. Thanks for the invitation. So when I look at the audience, so it's good to have a few people. So what I'd like to share today is I'm going to talk about some of the work in the past few years, primarily focus on the regulatory elements. In the past few years, why we need identify and annotate the regulatory elements called promoter or enhancer, silencer, and how these information can be utilized for the genetic selection on the complex traits in portrait. So before I started, so first I like to acknowledge, so there's so many of the people, so many of the scientists, in the US and in the world, collaborate in the past decades for my research program. So I really want to appreciate, so that's how I see the collaboration is everything for the research, particularly nowadays. Every lab only have a specific expertise. We need lots of people work together. My lab is primarily focused on the genetic improvement of the complex traits in portrait. So for the people in the audience probably familiar with called genome-wide association analysis. Basically, you get the genotyping and the phenotyping in a population, and then you're doing the statistical analysis and called a quantitative trait loci mapping, and then identify the genetic variants associated of the traits, complex traits, and then so you can do in the selective breeding and basically make a genetic improvement for those complex traits. So in the past many years, people always think about for the complex traits of genetic control, it's called a polygenic model. So talk about many genes control of the sort of the complex traits. In the past few years, there's a new study, a professor from Stanford, actually, he proposed called the omnigenetic model. So if you look at these figures, so you will see, so basically this omnigenetic model is talk about actually have a couple hundreds called core genes and contribute to the complex traits. And then actually we'll have more than 1,000, maybe 10,000 of these called peripheral genes in these surrounding area. Actually, they interact with the core genes. So that's how they contribute to genetic control of the complex traits. So in that sense, basically, the heritability is really spread extremely wide and across the genome as you expected. If you have 10,000 genes control complex traits basically everywhere, right? And then so the contributed variants, those genetic variants are highly enriched in the regions that active chromatin in the biological relevant tissue. That's how later on we'll talk about why we need to do the annotation, right? And then, so basically the idea is those couple of hundreds of core genes will have a direct effect, and then those thousands of those peripheral genes will act in trans. So what is the major challenge? If you think about it, have thousands or 10,000 of the genes control of the complex traits, basically we'll talk about we expect most of the case we were not able to find any major effect, unfortunately. So many of the time we find very subtle small genetic effect per genes. And then if we think about the number of individuals, right, and the statistical power, limit of statistical power will be very challenging to detect every possible of those genetic variants affect the complex traits. And another thing is basically later on you will see some of the examples. When you're doing the genome-wide association analysis, you will find many of the SNPs are significant, statistically significant, but which one is true, right? So we're talking about the correlation versus causality, right? The real causal genetic variance because of the linkage disagreement across the genome. And then as we realize those linkage disagreement will erode during the generation of the selection. So the one you selected works, a few generations later might not work because the linkage disagreement will disappear between the causal and the associated genetic markers. So how we can pinpoint those potential causal variants, then so you don't worry about this. Every time you select that genetic variant, you will expect to have a genetic impact on that. So basically, this is a talk about, for past decades, particularly in the biomedical field, when the people did the genome-wide association analysis with large population, talk about a million of those individuals. So they found more than 90% of those genetic associated SNPs are in non-coding region, not in the coding region, right? So basically, the idea is a variation in the gene expression is really a major factor contribute to phenotypic variation we observed for the complex traits. So this is the model basically talk about how those trait-associated genetic variants can contribute to complex traits. If you look at it here, basically this is a primary effect through these whatever transcription factor binding or the histone modification, right? Affect the probably contribute to the messenger RNA level, then contribute to the complex traits. And also can have a direct impact on the splicing or direct impact of the gene expression fine, or all of those contributed to complex traits. So if you think about the gene expression is a major contributor to the complex traits variation, so basically here give you idea about how we know the gene expression basically by promoter and more important the enhancer, right? So if you look at here, basically talk about for the promoter in general, it's pretty straightforward. So you can see the promoter usually is nearby transcription start site, that black. So you will see each of the genes has a promoter. But if you think about the enhancer, usually it's far away from the transcription start site from promoter. And then this is the red line, it's enhancer. You can see it's become more complicated because one gene could have multiple enhancers. Later we'll talk about some of the examples, how they regulate. Basically, the promoter initiate of the transcription, and then enhancer will be fine-tuned of the gene expressions. So here is a talk about those non-coding region, what kind of the genetic variants we are looking at here. So if you look at this enhancer region, you can see this is the transcription factor binding site. If there's anything of the mutation could change the transcription binding site. And that this is a variation basically can change the chromatin accessibility. And then from a promoter here, something can change the promoter binding. And then obviously it can change the splicing, and also can change the 3' end. So here give you idea about why those are, from a DNA sequence to finally of the complex traits, how this is related to the gene expression. So here just to give the example, we call it the context dependent, because here talk about between different tissues, right? And then those tissues, the genetic variants can affect different molecular phenotype, whatever DNA methylation, histone modification, ataxic gene expression protein, metabolites, microbiome, right? And then how each of these can contribute to different complex traits, right? Here I give the one of the example. Basically, it's that red line. Basically, talk about that genetic variants only affect that specific brain tissues, right? And then brain tissues, because the ataxic change affect the expression, so that expression affects the average DNA gain, right? So these genetic variants happened only changing in that specific tissue, not in any other tissues, right? Even with the genetic variation in other tissue detected, but not affect their molecular phenotype, will not affect their other phenotype. So that's why those tissue-specific regulatory elements are very important to identify and annotate them. So why FAN is a functional annotation of the animal genome. Basically, it's really from genetic variant to function in the complex traits. So first, we need to identify and functional annotate those regular elements across many different tissues, across different development stage, and also different physiological status. Then we can integrate all those functional information into the GWAS and to pinpoint of the potential causal variants. We'll give the example. And then once we identify these causal variants, we try to understand, as a biologist, we're interested in what are target genes of those genetic variants, particularly for those variants very far away from the target genes. And what is the molecular function of those causal variants, and how those causal variants regulate target genes, resulting in the complex traits variations. And here we're talking about from one dimension to a really multi-dimensional of those integration. So think about it from GWAS, we talk about DNA variants. Now we're talking about, so here basically give you an idea about these are the many different tissues in the different development stage, right? Here for these ones, talk about many different type of the molecular assays, epigenomic assays. You can generate a total different type of functional information because different tissues, different physiological states will have a different functional information, depends where are the genome of these. So this is the ENCODE, probably many of you guys are familiar with, it started about 2 decades ago. It's called encyclopedia of DNA elements. Basically, this is basically a gene have exon and intron. This is a promoter close to the transcription start site, and the enhancer far away, right? Those are all different type of high throughput epigenomic assay. We are able to basically provide functional information so we can identify and annotate of where are those promoter, where are those enhancer. I will give the example, a specific example of that. So here is a talk about, from a biomedical study, how they integrate all of those epigenomic assays. Obviously, human have so many of the samples, so many assays, so that how they work on, over 30,000 of the genetic loci integrated with 540 different complex traits, predict the trait-related that tissues, putative, causal genetic variants, and enriched of the tissue enhancer and candidate tissue-specific genes. So that's how complex of those information can be utilized by integrating this information. I know this slide is very, very busy. So basically a couple of years ago from Nature, basically from here, basically across many different tissues, cell line, and y-axis across all of those epigenomic assays, how they integrate all of the information with the GWAS, basically try to identify targeted genes for specific complex traits, identify potential causal genetic variants for specific disease. So that's how a few years ago, we are very fortunately leading that effort to focus on 3 major farm animal species. I put the chicken in the top. Obviously, chicken is my favorite one, favorite for all of you guys in the audience. So initially, we start with core A tissues with a very limited funding. So focus on those core assays. What we try to do is try to annotate these chromatin states and then identify, annotate of the regular elements, and also have all those resources available on the public database. So this paper published a few years ago in Nature Communications. You guys can find the detail of these. I will provide some summary of these. So basically, initially, so how we can give a summary of the epigenomic assay, right? What kind of assay we should use for this type of annotation for the farm animal genome community. So with the really good discussion with the biomedical community, so we come up with this RNA-seq, obviously it's molecular phenotyping, and then chromatin accessibility, and this is for histone modification. With the discussion, this is for the most informative of the epigenomic assay we can use to annotate with a limited budget. So as you can see, why we need to use 4 different histone modification marks. Basically, different marks provide a different activity of these, the promoter or enhancers or others regulatory elements. And a CDCF is a transcription factor. And then, so here just to give you an idea about once we connect all of the information across the 8 tissues, and then from all of those 5 assays, here you can see it depends on the combination of the different assays. So we use a chromatin hidden market model, so we can predict a total of 15 different, we call it chromatin states. So first 5, it's a different variety of the promoter, could be strong promoter, weak promoter, mediated promoter, and then 5 different of the enhancers and also other chromatin states. If you look at here, it's how many percentage of the genome of those regulatory elements are annotated across the 8 tissues. And then here, give you an idea about where are those regulatory elements located. If you look at the first of the promoter, you can see pretty much 9 very well, they are in the close to the transcription start site, 5' UTR, CBG island, for all of those, the promoter, make lots of sense. And then for the enhancer, as you guys can see, it's far away from transcription start site. That's why you didn't see any much of the enrichment from here. And then first is what we try to see. Once we identify those hundreds of thousands of those promoters and enhancer across different tissues, what do we look at here? Because we expect to have some tissue-specific activities. So we look at it here across the 3 different species. So here in the axis, the number of those tissues, if those promoters are shared across different tissues, if you look at it here, you will see across all the 3 species, so you will see many of those promoters that are shared across 8 tissues, right? Have a few of those promoters only specific in 1 tissue, some of only 2, 3, or 4, 5, right? Then if we look at the enhancer, so first we look at a genetic enhancer, which means those enhancers located inside of the gene body. And if you look at it here, you see total different pattern, right? So you see most of those enhancers acting in a tissue-specific manner. That's why this is very important for us to understand those are the tissue-specific enhancers play a very important role to regulate the gene expression. And then the second type of the enhancer, we call it endogenic enhancer because many of those enhancers really far away from the gene body, so they are in the endogenic region. So you see pretty much similar patterns, so they are really tissue-specific and not conserved across different species. A lot of the aspects of this from this type of study is it's basically we need to look at, particularly for the enhancer, where are those targeted genes, which targeted genes was the enhancer targeted. So this is involved called a 3-dimensional genomic organization, topological associated domain. If you look at it here, basically, it's talk about those enhancer, basically, even they're far away, so they can bend, form the loop, have a 3-dimensional interaction. That's how those enhancer can regulate the gene expression. And then that's how, from our study, we can use a CDCF, that transcription factor. And then basically, if we use this, we can predict the topology associated domain. And secondly, we can use H3K27 Annotation. It's active enhancer markers. So we look at the Spearman rank coordination across all of the gene expression. And then if there's a positive gene coordination, and then within the TAT, and then we can see, so basically that genes will be linked to that enhancer. So basically here provide an idea across the 3 species. For the blue bar is talk about if we just look at the nearby genes, right? And then talk about the prediction by our CDCF and the coordination. So you can see across all of the markers, first the 3 markers is the active marker. The last one is a repressor marker. So across all the border for genic enhancer, you can see our prediction is much, much higher than the nearby genes. And second, for the same thing for the endogenic enhancer, right? Look at the nearby genes and also look at our prediction again. So our prediction is to provide a much, much higher indicated. So many of the time, so the enhancer not really targeted the nearby genes, they're targeted far away off the genes. That's why those predictions are very important. So based on all those predictions, so that's how many of the TATs we identified. And that's if you look at it here, the genome coverage, what is the size of those TATs across the 3 different species? And then later on, so we're interested in extend from 8 core tissues across nearly all of the different type of tissues in the chicken genome. And then, so we're able to publish this paper last year in the Science Advances. And basically, we're talking about, we integrated more than 400, almost 400 genome-wide data assays and 23 tissues, annotate 1.6 million of the regulatory elements from 15 different cropping states, 1.2 million of the enhancer gene pairs, and then more than 7,000 of the super enhancers. So here, just to give an example how we annotate all of these. So if you look at it here, this is a CDH17, right? These genes only highly expressed in gut tissues, not expressed in any other tissues. If you look at it here, so this basically talk about the messenger RNA gene expression. You will see all the gut tissues have peaks, right? And then so we look at the H3K27 assonation, which is active enhancer. So you can see only those gut tissues have a peaks, not in any other tissues. And then we look at across many different assays. So you can see they're coordinated each other. All of those active assays have a peaks. But for the repressed marker H3K27 trimethylation, there's no peaks. So indicated how coordinated of different marks can provide information to help us to annotate all of these. So this is the enhancer gene pair prediction. So basically, if you look at it here, we talk about I have 5 different type of the enhancers. So this is how many pairs we predicted, how many of the enhancers involved, how many of the genes. If you look at the per vector per enhancer, how many of the genes? Roughly 3 genes per enhancer can target 3 genes. But if you look at the per genes, how many of the enhancers they can be targeted? So it's look at like 15 to 20, right? And then the distance from them usually is more than 250,000 base pair away. So it's very, very far away. So one of the interesting things we want to see, for some of the genes only targeted by few of the enhancers, but some of the genes targeted by many of the enhancers to see, so if the gene expression are different. If you look at it here, we divide it into the 3 different categories. The bottom one only targeted by 1 enhancer. The medium one is between 10 to 20 enhancers. And the top one basically targeted by more than 50 enhancers. So if you look at it here, you can see those are the one only targeted by 1 enhancer have a lower expression, but the one with targeted by many of the enhancer have a significantly higher of the gene expression. So next one, we want to look at if they're acting in a tissue-specific manner for those enhancer target, for those genes targeted by different type, many of the enhancers. So you will see those basically, those targeted by 1 enhancer, few of the enhancer, they are more tissue-specific. And then targeted by many of the enhancer, obviously they are more universal expressing other genes, other tissues. Then next one, we look at how conserved of those genes. So as you expected, those only targeted by single enhancer, they are more conserved versus, so they are less conserved, sorry. And then for these, targeted by many of the enhancers, so they are more conserved. So how we can apply all of this? So one of the things basically we're doing the selection signature to compare this is a Brunner mid-type of birds to Nair type of birds, egg-laying birds. So what we did, we look at the tissue-specific strong enhancer, just look at the strong enhancers. And then so different color indicated those tissue-specific strong enhancer. So if you look at it here, you will find a cortex, one type of the brain tissues, are significantly enriched in both of these, whatever Brunner and enhancer, and then the gut common of those strong enhancer. As you probably expected, the brain tissues, the gut tissues play a very important roles in terms of the genes related to the productivity, what of the laying eggs or meat productions. And then as we expected, the muscle-specific or strong enhancer are significantly enriched in Brunners and Genome significantly enriched in the layers. And then, so another area, basically, we talk about the functional annotation of the genome, right? When we're doing the genome-wide association analysis, so this is some of the significant SNPs. As you can see, for each of the region have many of those SNPs. Which of those are really causal? Basically, so that's how this chromatin activity we just talked about as functional annotation play a very important roles. Basically, here just give an example of 4 different tissues and look at those SNPs in this region, right? So for example, here you will see this is 3 SNPs. They have a functional activity in bone tissues, right? So indicated not other SNPs, but these 3 tissues might be potentially functionally important of the SNPs associated with biological relevant to the bone, maybe like bone density, right? So indicated how we can pinpoint and utilize those functional information to pinpoint from many of those significant of SNPs to narrow down small set of those SNPs. So here, just to give the example, so basically from the Brunner layers, this is a synaptic signature analysis. From here, you will see, here have the significance of the peaks here, not in the layer, right? And then we can zoom in to these regions, and this is our functional annotation, so you can see different color indicating of different chromatin states. The yellow color is a strong enhancer, so you can see in these, basically it's muscle tissues, have many of the strong enhancer in this. And then based on the enhancer gene pairs, so we can look at all of those enhancer paired with the genes to here called TAD4. And then we can look at the gene expression across many different tissues. You find that these genes basically only expressed in these muscle, heart, and adipose tissues. Suggest of these genes might be functionally important for the growth rate, and then this area of those SNPs in the enhancer might be play a very important roles to affect the gene expression, finally contribute to the growth rate. So we talk about the complex traits. How about the monogenic traits? This is a very unique, interesting of the example of these, the polydentity. So many of the people probably familiar with this, basically in terms of the multiple pores in the chicken and also in other mammals. So basically, this is just a single genetic mutation in the non-coding regions. So if you look at it here, this is from our annotation. If you look at it here, so this one located a strong enhancer in bone marrow, okay, in bone marrow. And then not in any others. And then if you look at how conserved of this look at across 77 vertebrates genome. This is a very, very conserved region. And that single mutation changes this. And if you look at these histone modification marks, you can see only in bone marrow have a strong peaks, not in any other tissues. And then interestingly, if you look at these specific mutation, this is in the mammal. So find basically, this is the long range of the enhancer, almost 1 million base pair away to regulate SHH genes. These genes for the limbud. So if you look at it here, this is all almost 1 million base pair away, and this is located in the intra region of this LMBR1, which is totally not relevant to this. And then if you look at it here, basically knock out this ZRS, their element, basically you will see their pores disappeared. And then again, if you look at it here from white type with these pores, human with these, and then fish have this, and then python, the snake, without this, and then knockout with this. And then again, for the human, you can see this is just a very single mutation in the intron, and then totally changes the enhancer, totally changes the phenotype. So as I mentioned, another area of the molecular phenotyping is expressor QTO. Basically, so here talk about if you're doing the gene expression for the thousands of the individual, right? So you can doing the eQTO mapping to find the different genotype that can affect the gene expression. How this information can integrate with the GWAS. That's you can do in the colocalization. Basically, if they're not colonized, maybe have a different mechanism to affect the complex traits. And if they're colocalized, which means that the genetic variants maybe affect regulated gene expression and also affect the complex traits. So that's how, like a few years ago, so with a collaboration from Lin Zhou Fan from Aarhus University in Denmark, so leading the Farm GTX initiative, basically called genotype tissue expression. This is a human biomedical field that started about 5 to 10 years ago. So basically, we utilize all of those public database, roughly 2,900 of those whole genome sequences, roughly more than 7,000 of RNA-seq data across all different breeds and different tissues. So here, just to provide you a idea about in each of those tissues, how many of those RNA-seq are available. As you can see, some of the popular of the tissues like liver have almost 800, right? Spleen have almost 400, but some of them have a very limited, but minimum we need at least 50 of those RNA-seq data in order to doing this type of analysis. So what we did, we basically look at the 5 different molecular phenotypes. So we look at the expressing QTO linker RNA QTO, basically the average of those gene expression, right? Look at the SNP affect those gene expression. Actually, we can look at the exon, which look at only first exon, right? Exon QTO. And then we can look at the splicing QTO. And then also we can look at the 3' A QTO, look at the point A of those QTO. Here, just to give the example, so basically this is a expressed QTO we identify in that spectral region in the brain tissues, right? If you look at that spectral region, this region exactly located in here. This is by our functional notation. As you probably see, this is the yellow color again. It's a strong enhancers and across those 3 main brain tissues. So make lots of sense. This is the EQTO in the brain tissues, and also it's a strong enhancer region across those 3 brain tissues. Then we're doing the co-localization. So basically what we did, we look at almost 40 different complex traits in advanced intercross population. So we use 4 different models, different models have a different hypothesis. Basically try to look at how many of those GWAS associated SNP will be co-localized with those molecular QTO. As you can see, some of these overlap, some of very limited overlap. In general, we find only 30–80% of the GWAS nuclei could be linked to at least one of the molecular QTO. As you can see, most of these are not linked to any of those QTO, indicated one. So there's a different molecular mechanism affect the complex traits. Secondly, we still need more of the data in order to identify those expressed QTO. So here, just to give the example, so when we did called the TWAS, the transcriptome-wide association analysis, we are able to identify functional genes. So in this case, we work on this average DNA gain for week 6 to 8 in birds. And then we find that this KPANA3 called carotene submitted alpha-3. So if you can see these genes basically have 2 of those expressed QTO in this. And if we look at the expression of this, we find that the retina is significantly enriched. And then next one is the pituitary gland, okay? And then we look at it further, we find that this leading SNP, so about 10 possible of the causal mutation across all tissues, but this one, this one is the most significant one. If we look at it here by the GWAS study, this is not the top SNP, but it's a leading SNP, it's a significant SNP here related to average DNA gain 6 to 8 weeks. And then we look at the co-localization, we find that the retina expressed QTO and the pituitary splice QTO are significantly reached. And then if we look at this, we find that these 2 have a high linkage discriminant, indicated both of these that have the same mechanism of regulate of those complex traits, versus the other one with a very low linkage discriminant indicated suggests the potential 2 potential causative variants. So last example, I like to give one of a specific example on the other technique we can utilize. So people are familiar with COP700 and a random mating of birds. You can see, if you look at the body weight, you can see significant difference across the 4 weeks of the age. When we raise this together, we look at their promoter in the muscle tissues and 4 weeks. So you will see across all of those individuals, so they have a strong promoter. But if you look at the enhancer, you will find only enhancer appear in the COP birds, not in the ACRB birds. If we look at that specific region, we find they have a total different scale of the alleles. So for the COP, 86% of the birds, because we have almost 50 birds from that population, 50 birds from ACRB. So you can see totally skewed from one allele versus another allele. And then if we look at that specific mutations, we find through our 3-dimensional structure, we find for the cup birds with that allele have loop interaction versus this here. So basically without the loop interaction. So the single mutation can change the 3-dimensional interaction, potentially can change the enhancers. So from that resource, basically the genome browser can provide all of those chromatin states across all of these, and then you can check all of those tissues. And then, so this is the Chicken GTAX portal. So you can look at all of those expressing QTOs, and then it's going to here give the one of the example toll-like receptor file. You can look at all of the gene expression across different tissues. Where are those expressed QTO? And then other 3' and QTO. And in summary, so tissue-specific regular elements are very important to contribute to genetic variation of the complex traits, and really integrate all of the omic data can accelerate potential causative genetic variants. And then I do believe it's a great time to bridge genome to phenome, and this portal really provide a huge resource for the scientific community. Thanks very much for all your attention.